All right, hi everybody. Good afternoon and welcome to today's webinar sponsored by NAVBO's Education Committee. I'm Josh Scallon from the University of South Florida, and I'll be moderating today's session. I'm pleased to welcome our speakers, Dr. Courtney Griffin from the Oklahoma Medical Research Foundation and Dr. Cindy St. Hilaire from the University of Pittsburgh. Before we begin, I'd like to remind you that all NAVBO activities are now easily found in the calendar of events on our website. Please be sure to register for some of these upcoming events. Throughout each presentation, please type your questions in the question box. These questions will be answered at the end of each talk, but you don't need to wait until the end to type in questions, so please type in questions throughout. This webinar is being recorded and archived on the NAVBO website for future use. First, I'd like to introduce Courtney Griffin. She'll be presenting her talk from the IVBM entitled, What Chromatin Remodelers Can Teach Us About Vascular Development and Integrity. Courtney Griffin was raised in Athens, Georgia. She earned her BA in biology at Harvard and her PhD in biomedical sciences at UCSF with Sean Coughlin. She then pursued postdoctoral training in genetics with Terry Magnuson at UNC Chapel Hill. In 2008, she launched her own lab at the Oklahoma Medical Research Foundation, studying blood vessel development and maintenance in health and disease. Courtney is now a full member in the Cardiovascular Biology Research Program and Vice President of Research at OMRF. She is also the immediate past president of NAVBO and a standing member of the NIH CDD study section. So with that, welcome Courtney, and feel free to take over.
All right, thank you, Josh, and thanks to everyone for being here. I'm really pleased to have this opportunity today to talk to you about an approach that my lab uses to really identify and study genes that are important for vascular development and integrity. And this approach really focuses on exploiting chromatin remodeling complexes. And specifically, we focus on these ATP-dependent chromatin remodeling complexes, which are large multi-protein complexes. They contain as a central catalytic subunit an ATPase, as the name implies. And this ATPase can generate energy to help these complexes break DNA and histone bonds and thereby start to evict and slide nucleosomes in these compacted chromatin regions to lead to their decompaction. And when that happens in a gene regulatory region like a promoter, as is shown here, it can result in either the increase or decrease in transcription of a target gene because it allows for the recruitment of activator and repressor proteins and complexes which had previously been unable to access DNA in these compacted chromatin regions. Now they can access it and they can impart their effect on transcription of target genes. So because, as I said, we're interested in genes that regulate the vasculature, we decided to exploit these complexes, which tweak transcription, to try to identify some critical genes. And we do this by using genetic deletion of the ATPase of these complexes using Cree-Lox technology. We knock out the ATPase in vascular cells like an endothelial cell. And then we really focus in on the animal and look for a really highly penetrant, consistent vascular phenotype. Because if we see something like that, it tells us that we have transcriptionally impacted a consequential target gene in the mutant endothelial cell. And now there's either too much or too little transcription of that target gene. And that is presumably what's leading to the vascular phenotype. So, a couple of notes about this approach. You might say to yourself, well, surely if you knock out one of these major remodeling complexes, you're going to have so many transcriptional impact on the genome, so much transcriptional impact that you would never be able to correlate a specific target gene to a vascular phenotype. How would that be possible? And I just want to point out that, these are not transcription factors. And so the changes that you get when you knock these complexes out are fairly subtle. You don't get massive changes in transcription. You get a lot of changes, but most of them are fairly modest. And most genes, frankly, are pretty refractory to these small transcriptional changes. So what this approach really does highlight, though, is those few genes that require really tight transcriptional regulation. And if you misregulate it, it can have a negative impact on the vasculature. And so how do we prove then that we've identified a target gene that is causing a vascular phenotype? Well, we take either genetic or pharmacological approaches to manipulate a candidate target gene when we think we have one in mind, and then try to rescue the vascular phenotype that we generated in our chromatin remodeling mutant, and that then would tell us that we've hit on the right target gene for the right phenotype. And so today I'm going to talk to you about one such gene that emerged from using this approach in our lab, and this is called RIPK3. You'll sometimes hear me call it RIP3, and this is the receptor interacting protein kinase 3. And we stumbled into RIP3 when we were studying the NERD chromatin remodeling complex, which has as a central catalytic subunit CHD4 as an ATPase. We had a floxed allele for CHD4, and when we knocked it out using the Tie2 Cre, which many of you know is expressed early in development, comes on around embryonic day 7.5 in endothelial and hematopoietic cells. And we were really excited to see just a fantastic phenotype in that all of the mutant embryos were dead by embryonic day 11.5 with a really consistent phenotype. And that was a phenotype of this massive and very sudden vascular rupture that happened here in the mid region of the embryo. So these embryos looked really good up until about E11, and then within half a day, they all looked like this. And if we backed up a day to E10.5, again, the mutant embryos were totally indistinguishable by eye from the controls. But if you looked at the rupture-prone vasculature here in the mid-region, you start to see some morphological abnormalities in the endothelial cells. So here in these semi-thin sections, you can see the endothelial cells are kind of swollen. And then by transmission EM, you can see even that the organelles are swollen and the plasma membrane is starting to break down, as you see in this inset. So these are all hallmarks of necrosis, which we thought of as a really kind of non-specific sort of death, and we had no idea why knocking out a chromatin remodeler might be leading to this necrotic-looking morphology. And that was until we performed some microarrays on our control and mutant embryos. So this was many years ago. We backed up even a day prior to this electron microscopy. We backed up to E9.5. We did not see many differentially expressed genes at this time point, but one gene that was differentially expressed was RIPK3, and it was, modestly upregulated, 1.23-fold, but it really jumped out to us because at that time, RIPK3 was starting to be described as being involved in a programmed cell death pathway called necroptosis. So this is a programmed form of necrosis. And given these necrotic phenotypes we were seeing in our mutant endothelial cells, we said, this might make sense. And specifically, we were hypothesizing that elevation of this RIPK3 in the endothelial cells could potentially be mediating this particular type of cell death, this necroptosis. And that would then be making this vasculature here in the mid-region of the embryo weak and fragile and prone to rupture. And so, sure enough, we were able to show that this candidate target gene of interest, RIPK3, that it did indeed contribute to the vascular rupture in our mutant embryos. So just like I told you in the intro slide, this is a genetic approach we took to prove that we were on to the right target gene. So we knew we had too much RIP3 and we wanted to reduce it. So we used either a global allele and knocked out one copy of it. We also made a floxed allele of RIP3, and so we knocked out one copy of it in endothelial cells. In both cases, we were able to rescue that massive vascular rupture that had happened in the mid-region at E11.5. Now, notice here we're looking at E12.5, so all the mutants are dead by this point, and they've been dead for a day, so they're partially resorbed. But you can see here that these mutants in which we reduced a copy of RIP3, they had survived that lethal vascular rupture at 11.5. Now, they were starting to develop some head bleeding, but at least they had avoided this mid-region bleeding. So we very logically and simplistically assumed that the CHD4 NERD complexes were in the business of repressing RIP3 transcription in endothelial cells, and that if you knocked out CHD4 in endothelial cells, you would get too much RIPK3, which led to this vascular rupture. Turns out that was not quite what was going on, at least not under basal conditions, and it took us a while to figure this out. But it is true that CHD4 suppresses endothelial RIP3 transcription, but it does it specifically under hypoxic conditions. And so normally under normoxia, there's a basal amount of RIP3 that's transcribed in endothelial cells, and CHD4 doesn't care about that. It doesn't get involved in that. Where the CHD4 complex comes in is under conditions of hypoxia. And I just want to point out that mid-gestation is a hypoxic time for a developing embryo, because it has been relying on the yolk sac up until about E10 for an exchange of oxygen between the mom and the embryo, but then eventually that's not sufficient, and it needs to hook up, the embryo needs to hook up to the placenta to get sufficient oxygen to sustain its development. And so right around 10.5, the embryo is sort of at peak hypoxia, and this is where the CHD4 NERD complex becomes important in terms of putting some brakes onto the RIP3 transcription so that you don't make too much of it. So, in our case, when we had genetically deleted CHD4, sure enough, the RIP3 promoter became permissively decorated with acetylation sites, the transcription shot up, and that's what ended up killing these embryos because they had too much RIP3 in their endothelial cells. Now, there were a couple of surprises that actually came out of this story. Number one, our hypothesis was not quite accurate. Yes, it was true that we had too much RIP3, and yes, it was true that this was causing vascular rupture, but it turns out it wasn't happening as we suspected through the necroptosis pathway. And we knew that because we could knock out this downstream target of RIPK3 called MLKL, which is really critical for the necroptosis execution. If we knocked out MLKL in our CHD4 mutants, it did not rescue the embryos, unlike when we knocked out RIP3, and we did get that rescue. So this tells us that RIP3 is doing something else in these endothelial cells to cause this vascular rupture, and that's still a bit of a mystery. Another surprise that came out of this work is the fact that at this mid-gestation stage of development, there's actually quite a bit of RIP3 expressed. This is a wild type embryo published by another lab, and they're showing that there's actually quite a lot of RIP3 in the vasculature, in the endothelium of these embryos. And so it started making us think, okay, so maybe if there is all this RIP3, maybe it's not a bad guy. Obviously, these embryos, these wild type embryos go on and survive. So does RIP3 actually have some beneficial roles that have been unappreciated? And I had a couple of wonderful grad students over the past several years, both of whom are still heavily involved in NAVBO now that they're in their postdoc labs. And they both showed that, yes, RIP3 actually does have some interesting and sort of unexpected beneficial roles in the vasculature. So Sarah showed that it's actually atheroprotective in an atherosclerosis model. And specifically, RIP3 protects against plaque accumulation in the descending aorta, a region where usually you don't have plaque buildup. But if you knock out RIP3 using our FLOX allele in the vasculature, in the endothelial cells, you start to get plaque accumulation in this descending aorta. And then Sichi showed that RIP3 actually helps promote, normal developmental angiogenesis, both in the embryo, and she could see this in the brain, but also in the postnatal retina, which so many of us study. So RIP3 has this surprising pro-angiogenic role as well. And I think these two stories, which I don't have time to go into, really help to start to shift the conversation about RIP3. And they demonstrate that this is one of those genes that I talked about in the very intro slide, where it needs to be tightly transcriptionally regulated in an endothelial cell, because either too much of it or too little of it, as these two people showed, can be very undesirable in an endothelial cell. So I want to spend the remainder of my time telling you about an unpublished study that's being led by a postdoc in the lab, Charmaine Johnson. And Charmaine is really interested in whether endothelial RIP3 is beneficial or detrimental during a specific type of injury, and this is ischemia reperfusion injury. And we thought this was a relevant question because, of course, as this model implies, ischemia causes hypoxia, and the embryonic work we had done showed that RIP3 is elevated under hypoxic conditions. We also knew from the embryo that too much RIP3 has negative effects on vascular integrity. It causes rupture, it causes vascular permeability. And so Charmaine was interested in whether these different paradigms would also hold up in a postnatal context. And so she introduced this ischemia reperfusion, or IR injury model, into the lab. It's a surgical model. She focuses on the mesentery, and she's able to clamp these first order mesenteric arteries coming off of the superior mesenteric artery, and thereby block blood flow to the downstream intestinal tissue. And so you get these ischemic regions downstream of the clamp. And notice that Charmaine also leaves some unclamped arteries as well, and those become control regions, and we consider them proximal control regions if they're kind of close to the clamp, and then more distal control regions if they're further away. And Charmaine leaves the clamp on for 1 hour, and then she allows the tissues to reperfuse for 24 hours. So the first thing she asked was, does this injury cause an elevation of RIP3? And to look at this, she did some really beautiful en face imaging of these arteries downstream of the clamp in this ischemic region. And using a RIP3 reporter mouse, you can see here that even after just 1 hour with the clamp, yes, there's an elevation of RIP3. And even 24 hours later, after the reperfusion, RIP3 is still elevated in these ischemic vessels that had been downstream of the clamp. So the next question then was, well, what is that elevated RIP3 doing? Is it beneficial? Is it detrimental? How's it impacting vascular integrity? And she decided to look at this using an Evans blue dye extravasation assay. This is where you circulate this small dye, which usually stays in the vasculature, but if it leaks out, that tells you that there's increased vascular permeability. And she took advantage of our RIP3 floxed animals, this time crossing them onto Ralph Adams' inducible CDH5 Cree to knock them out, to knock out RIP3 in the endothelium. Charmaine happened to induce these at 8 weeks of age, and then she waited 4 full weeks for full deletion of RIP3 before doing the IR injury. And so the first thing that she noticed was not a surprise from the literature. She looked at Evans blue leakage in this ischemic region of the gut, downstream of the clamp. And as expected, yes, there is leakage even in control animals downstream of the clamp, and that was true also in the mutant animals. What became a surprise is that even in the proximal control regions and even the distal control regions further away from the clamp site, only in the mutants did she continue to see elevated vascular permeability. And not only that, even in further away remote organs like the lung, the liver, and the kidney, although interestingly not all organs, for example, not the heart, she saw elevated vascular permeability in the mutant animals. And notice, this did not happen in uninjured animals. This was only after the IR injury. So it made us start thinking about what is it that could be circulating away from this clamp site that could be elevating the permeability in these far away sites, organs, and vasculature. And of course, we thought about inflammatory cytokines. and she did a panel of ELISAs looking at different cytokines, and noticed that in the injured mutant animals, there was an elevation of IL-6, both in the ileum tissue and in the circulating serum. And IL-6 is one of these inflammatory cytokines that is associated with vascular permeability. She at this point was curious to know where it was coming from. It would make sense that it might come from blood, but she had not mutated the blood. And keep in mind that she used a lot of really elegant reporters and qPCR analyses to really verify that she had only knocked out RIP3 in endothelial cells using this CRE, and it was not being excised in blood cells that typically make a lot of these cytokines. And so then further evidence that this elevated IL-6 was coming from the mutant endothelium came from in vitro experiments. So we have this murine MS1 endothelial cell line in which we deleted RIP3 using CRISPR-Cas9. And if she challenges these cultured cells with TNF alpha to simulate, an inflammatory injury like the mice had received, here too she sees an elevation of inflammatory pro-permeability cytokines, including IL-6, which she had seen in vivo, and then IL-1 beta. On top of that, Charmaine also saw some really interesting elevation of adhesion molecules, both in vivo and in vitro. And this is at the protein level. She saw VCAM elevating in the ileum and ICAM specifically in the lung. And the same was true in the cultured mutant cells. And so from what I've told you, it looks like endothelial RIP3 is really protecting against vascular permeability, an elevation of inflammatory cytokines, and an elevation of adhesion molecules, specifically in an injury context. The story, though, does kind of extend beyond the endothelium in the sense that it does involve some crosstalk with circulating blood. So she was able to suss this out by doing a monocyte depletion in her mutant animals using claudronate liposomes. And when Charmaine depleted the monocytes and then did the IR injury model, she saw that vascular permeability was actually rescued in these animals. And it was rescued specifically in the ileum and in the lung. And very interestingly, those were exactly the two sites where we had seen adhesion molecules upregulated, ICAM and VCAM. And so altogether, to synthesize this, what it's telling us is that endothelial RIP3 is preventing vascular leakage during IR injury. And if you knock it out, you're getting an elevation of local inflammatory cytokines from the endothelium that are probably contributing to some of the vascular leakage. But you're also getting this elevation of adhesion molecules in a very organotypic fashion. And that's allowing, of course, for circulating monocytes to get stuck to this mutant endothelium. And there, too, I'm sure they're releasing inflammatory cytokines and they're extravasating, causing even further vascular leakage. So it's a really cool crosstalk story that is focused back on this endothelial cell molecule that we knew nothing about when we started this process of mutating chromatin remodelers many years ago, but we've been delighted at how this approach has achieved exactly what we hoped, in that it opened our eyes to a novel gene that has interesting roles in the vasculature, and I think we're just at the tip of the iceberg in terms of what we have to learn about RIPK3. So I'm going to end there and acknowledge the people currently in the lab, as well as a few people that I mentioned who are former lab members, our current funding, and people who donated mice to us, which we're super grateful for. So thank you, and I'm happy in the last couple of minutes of my time to answer any questions.
All right, thank you, Courtney. That was an excellent presentation. While we wait on questions to fill in the question box, I had some kind of general questions for you about your talk as well. mean, I kind of thought about this question in the first part and the second part, so it's kind of hits on both things. But when you mentioned the definition of RIP3K, right? It's a receptor interacting protein, which means maybe it interacts with a whole bunch of other proteins and receptors inside the cell. So have you done any kind of large screening, maybe like a bio ID kind of experiment to figure out what all proteins that RIP3K interacts with, and whether or not, you know, if you did RNA-seq with cells treated with TNF alpha, whether or not you could identify specific inflammatory cytokines that it regulates?
Yeah, it's a really interesting question. There's so many things we could do, Josh. You're right with this protein. So we did do some IP mass specs. This was back when Sichi was studying kind of this interesting pro-angiogenic role for RIP3, and that actually sort of opened up a mechanism related to angiogenesis that involves an interesting molecule called myofurlin, which Bill Sessa's lab had identified many years ago to regulate VEGF receptor 2 expression at the cell surface. And so RIP3 is playing with myofurlin in that context. It also revealed, and we still don't understand the full context of it, that RIP3 interacts with a lot of RAB proteins, so proteins involved in the endocytic pathway and in vesicles. And so we think RIP3 has some interesting trafficking roles in the endothelium that are yet to be fully realized. And whether those impact some of the IR study will be interesting to see. But it is powerful having, you know, at least these CRISPR mutated cells to work with, and, you know, the more tools that we have to sort of suss out roles for RIP3 in the vasculature, I think the more we'll learn.
Yeah, definitely. That's very exciting. And exciting for me to hear that it interacts with RAB proteins, because they regulate weak adherent recycling, right? So
yeah, good point. And again, something in terms of something important for, you know, vascular integrity, I think that's really that's a cool thing to think about, especially hearkening back to the embryo studies where we don't really know what too much RIP3 was doing in that context, because we know it's not necroptosis.
It's pretty cool. Okay, so we have 2 questions. One question is from Allison Dubner. Do you know if anyone has looked at RIPK3 and other vascular cells like smooth muscle cells?
Oh yeah, good question. So yes, people have, there's actually been quite a lot of work focused on the vasculature, but using global RIP3 knockout animals. And that's revealed some interesting biology. What I find most fascinating as an endothelial person is that there's somewhat of a disconnect between what the global knockouts of RIP3 tell you in some of these disease models. Usually it's telling you RIP3 is a bad guy and you want to get rid of it, versus these cell-specific roles that we've seen where RIP3 can be quite beneficial in the endothelium. And so I think there's a lot of interesting basic biology to be learned from that, including in smooth muscle cells. But it's also raises some cautions about using kind of broad spectrum anti-necroptosis pharmacological agents, you know, to treat a disease that may have subtleties in terms of the cell specific context for something like RIP3. And I think so it's sort of my passion plea for, we need to understand more about cell specific roles for genes like RIP3 before we sort of take these broad approaches toward deleting it everywhere. And perhaps by knowing cell specific roles, we can develop better, more targeted therapeutics for some of these diseases that it plays in.
All right. So while we were talking, several more questions appeared in the question box, but it looks like we're running a little bit short on time. So I'm going to encourage everybody to email those to Courtney, or if Bernadette could email those to you, then you can respond one by one.
I'd be happy to. Thank you.
Thank you very much. Okay, so now we're going to introduce Dr. Cindy St. Hilaire, who's going to present her talk entitled Novel Mechanisms Driving Vessel and Valve Calcification. Dr. St. Hilaire is an assistant professor of medicine and bioengineering at the University of Pittsburgh and a faculty scholar at the Vascular Medicine Institute. Dr. St. Hilaire obtained a BS in molecular genetics from the University of Vermont, a PhD in biochemistry at Boston University School of Medicine, and a postdoc fellowship at the NHLBI at the NIH. At the NHLBI, Dr. St. Hilaire and colleagues discovered the genetic cause of the rare disease, arterial calcification, due to deficiency of CD73, which marked the start of her research in the field of cardiovascular calcification. Her research in the lab at the University of Pittsburgh focuses on identifying and characterizing the mechanisms underlying the development of vascular and valvular calcification pathologies, with specific interest in defining the mechanisms by which inflammation and mechanical stress drive the transformation of a healthy vascular cell into a calcifying cell. Dr. St. Hilaire is an internationally recognized expert in the field of cardiovascular calcification. She's an associate editor at Circulation Research and is the host of the monthly podcast Discover Research. Welcome, Cindy, and feel free to take it away. I think you're muted.
Almost forgot to unmute. Thank you so much for this opportunity and the invitation to present my work to you today. I'm going to be talking about our work on valve calcification, and so I do have one disclosure that is related to that. And so I guess just a brief overview, my lab is interested kind of in all aspects related to cardiovascular calcification. What initiates calcification, meaning what are those initial stresses that can trigger a cell to start to become unhealthy? What's going on inside the cell? So what's modulating that osteogenic transition? What's happening at the level of the chromatin, what's happening at the level of activation or repression of specific transcriptional programs? And then lastly, what's promoting the propagation of that cardiovascular calcification? And ultimately, what can we do to stop it? And so I'm going to be talking today about calcific aortic valve disease. And so these are two specimens that my lab received. So we, the word translational science it's a very wobbly definition. We use it to mean that we start with investigations in the human. So working with our donor program here at the University of Pittsburgh, but also working very closely with surgeon scientists, we have a very large biobank now of both control and diseased calcified valve tissue. And this is kind of the general progression of calcific aortic valve disease. You have a normal leaflet that starts to remodel. Those leaflets get a little stiff, a little thick, and calcification starts to precipitate in the leaflet. This is a bit of a challenging disease to study, mostly because mice don't readily recapitulate this disease. When we see calcification in the, quote, valve of a mouse, it's usually in the aortic root. We don't really see a ton of the leaflet calcification, which is predominantly what we see in a human, and that's what makes this difficult. We have to be really careful about what questions we're asking in a mouse versus in a human. And then ultimately, because of these complications, there is no therapeutic option for calcified valves. You're looking at the two options, and that is replacement. So you have to wait for your valve to get diseased enough to start to cause damage to the heart, and you would either get a replacement with a mechanical valve, which you will then have to be on antithrombotic or antiplatelet therapies forever, or you get a bioprosthetic valve, which these currently only last about 7 to 10 years. And I'm not going to get into how these get implanted per se, but suffice it to say, it is no walk in the park. These are highly invasive, highly, risky, depending on what other comorbidities you've had. And ultimately, what we know about valve calcification and also vascular calcification, it starts with what we know from bone. And so in bone, you have a cell called the osteoblast. And the osteoblast has a very specific transcriptional program. One of the things that drives a stem cell to become an osteoblast is a transcription factor called RUNX2. It's considered the master regulator of the osteogenic transcriptional program. And an osteoblast will secrete enzymes and also the minerals that create the minerals, but also enzymes that help to create those minerals that will be excreted or kind of released in matrix vesicles that accumulate in the extracellular matrix space. And that's how you get bone remodeling. Bone is happening outside of the cells. Similarly, arterial calcification and also valve calcification has very similar processes at play. You know, we have extracellular matrix degradation contribute to calcification. When matrix vesicles accumulate in that space, phosphate and calcium imbalance can nucleate in these damaged matrix compartments. Apoptosis and inflammation are some of those initiating stresses that help drive a cell to transition into a calcifying cell. And then we have failed anti-calcific effects and the active osteogenic differentiation and genetics also playing a role. And so, again, my approach to studying calcific aortic valve disease really started with asking the question, how is it that a healthy cell is turning into an osteogenic, a calcifying cell, right? And one of our questions is, you know, does this healthy valve interstitial cell just have to upregulate a key gene, maybe alkaline phosphatase, which can allow for calcification particles to be made, the building blocks? Or does this healthy cell kind of undergo a drastic reprogramming, chromatin remodeling, like Courtney was talking about, whereby it will start to actively repress and then subsequently activate a new transcriptional program? And so when we started to think about this, we went back to the cells that make vascular cells, and that is the mesenchymal stem cell. So the mesenchymal stem cell, I'm sure a lot of people understand, can become many different types of cells. And mesodermal cells include the connective tissue, cartilage, and bone, but a mesenchymal stem cell can also become a smooth muscle cell, right? And so this mesenchymal stem cell is kind of a progenitor of a lot of the tissue that we are interested in that can calcify. And so when we were doing some reading, trying to understand more about what is a mesenchymal stem cell and what are the triggers that cause it to become X or Y cell, we came across these two papers. These are from 2002, and that was right when TERT, or telomerase complex, were really starting to become interesting and in the news a lot. And people were using the overexpression of the gene, or TERT, to drive the immortalization of cells. The idea being that if a cell divides and divides but cannot or does not have telomere attrition, it can live forever. So these people, these two independent groups, overexpressed TERT in a mesenchymal stem cell, human mesenchymal stem cell, and they found that when they did that, the mesenchymal stem cell differentiated into bone-forming cells. And you can see here, this is actual bone with the little, osteocyte signatures. And you can see very nice hydroxyapatite stain, which is the mineral form of the bone. And so what we know now, that was a 2022 study, so 11 years later, we know that yes, TERT does have what we call the canonical telomere extending functions, and that's where it combined with TERK with the C to provide the cap extensions on telomeres. But TERT has non-canonical transcriptional regulatory roles. TERT can interact with chromatin remodeling proteins and also directly with transcription factors to drive the access and then the upregulation of different genes. And so we started to ask the question, does TERT promote the osteogenic transition of vascular cells? We know that it played a role by overexpression in mesenchymal stem cell, but what's happening in a differentiated cell? And so again, we started by looking at the tissue. So we went to our tissue bank and we started to look at specimens from control and CAVD tissue. And we could see that in the calcified tissue, which is the blackish stain here, this is just a black and white image, but in that tissue, serial sections, we could see a drastic upregulation of TERT in those tissues. And this was independent of any changes in the telomere length of those tissues. We also wanted to start to look at this in the dish. And so we just directly isolated cells from these control and calcified valve tissues. And when we looked at these cells just at baseline, we didn't see any differences in proliferation or the migration potential, but we did see at baseline, and these are all representing individual lines from individual patients, a baseline upregulation of TERT and a baseline upregulation of that osteogenic master regulator RUNX2. And so one of the things that we were able to do in making this biobank is develop a really great way to preserve these tissues when they come from the clinic or when they come from a donor. They get put in a very special solution right away that allows us to process these cells for up to 48 hours after extraction, and we're still able to get viable cells. And this is the cells that we use for our in vitro disease modeling. And so one of the things we did was to see if we knocked down TERT, if we could prevent calcification. And in a dish, we could. So in a VIC put under osteogenic conditions, and that's the media that can differentiate a stem cell into an osteoblast-like cell. When we put control shRNA in, there's no difference. Our cells very nicely calcify at 21 and then at 28 days. And if we use shRNA to knock down TERT in these cells, we see a very drastic reduction of calcification at 21 and 28 days. We know that this suppression of calcification without TERT is not due to the telomere extending activities of TERT. If we use the TERT enzymatic inhibitor BIBR 1532, we see no evidence of changes in calcification. We also don't see any changes in the expression of TERT or some of the downstream genes that we're interested in. We also wanted to see if this was just specific to valve cells or if it happens in other cells as well. And so we did the same experiment in human coronary artery smooth muscle cells, and we also did it in human mesenchymal stem cells. We also used genetic knockout cells from mice. We used, again, the valve interstitial cells or the bone marrow mesenchymal stem cells. Essentially, thus far, in every cell type we've looked at that can calcify, so VICs, coronary artery smooth muscle cells, and mesenchymal stem cells, if we knock down TERT, we see a drastic decrease in calcification, which is really exciting. But what the heck's going on there? Why would a cell that is triggered to calcify have anything to do with TERT? What else is going on there? And so to think about what might be inducing TERT and perhaps other proteins to contribute, we went back to known diseases, human diseases, where individuals present with early calcified valves. And so people who have chronic inflammation, whether it's from endocarditis. So a lot of IV drug users get blood infections and they can get endocarditis, and they will develop severely calcified valves. People with rheumatic heart disease also have early presentation of calcified valves. A lot of times it's the mitral valve, but you will also get it in the aortic valve. And then patients with interferonopathies. So those are genetic diseases with activating mutations that trigger hyper immune interferon signaling in their body. And we also know from some nice studies in mouse and also in porcine VICs that interferon signaling does play a role in calcification. And so we know that TERT has non-canonical effects, but TERT itself does not bind DNA. TERT can be an adapter, it can be a scaffold, it can bring chromatin remodeling structures and transcription factors together, but it doesn't bind DNA itself. And so we then took a step back and thought, well, we know RUNX2 is up in our cells, but what is actually driving its expression? And so we kind of just did an in silico screen and found that there were a relatively large number of STAT5, putative STAT5 binding sites on the RUNX2 gene promoter. And when we looked at our tissues, at our cells from tissues under no treatment or under these osteogenic treatments, we could see that the STAT5 transcription factor is up at baseline. And it's important to know that STATs in general, and also specifically STAT5, are under the regulation of interferon signaling and other inflammatory signaling markers. When we had control cells from control valve cells put under no treatment or osteogenic conditions, we could see a handful of transcription factors and proteins upregulated that had to do with osteogenesis and VICT activation and cell remodeling. And we did see that STAT5, one of the isoforms, was upregulated in these tissues. So we want to see, is this perhaps one of the transcription factors playing a role in the calcification? And so we could see that in cells under osteogenic conditions, we do see an increase in STAT5 nuclear localization. You can almost see in the no treatment, you know, that STAT is localized to the cell membrane where it's not active, but then in the OST conditions, it's not really localized on the membrane. It seems to be really in the nucleus. And so we asked the question, are TERT and STAT5 interacting in the cell when that cell is under osteogenic stimulation? And so we performed a proximity ligation assay. It's where you can use 2 antibodies that then have 2 other antibodies that have little pieces of DNA that can serve as primers. And if you see amplification, this essentially is qPCR on a slide, and these red puncta indicate that TERT and STAT5 are in close proximity. And we can see that when these cells are put under osteogenic stimulation, this is an assay that was assessed at 2 weeks, we can see TERT and STAT5 highly co-localized in the cell. We're also able to do an immunoprecipitation assay where we pull down STAT5 and Western blot for TERT, and we can see that indeed STAT5 does pull down TERT under osteogenic conditions, but not under the no treatment conditions. This is really exciting. And so we then wanted to assess by chromatin immunoprecipitation whether TERT or STAT5 does bind to these putative binding sites in the RUNX2 promoter. And indeed, that's exactly what we see. When we pull out STAT5, we are able to see binding at 2 different putative STAT5 binding sites in the RUNX2 promoter. We also took an unbiased approach using cut and tag, and again saw these 2 sites as positive hits for STAT5 binding. So this is great, but you know, we can't really leverage TERT for therapy itself, right? We don't have the enzyme inhibitor didn't block activity, and you know, we don't want to, like, you know, delete TERT from a cell because it'll age it. But maybe we can harness the fact that it's interacting with STAT5. And so in our in vitro calcification assay, we gave a STAT5 inhibitor, one of 2 different ones, and in valve interstitial cells and also human coronary smooth muscle cells, inhibiting STAT really drastically reduced calcification in our in vitro disease model. We also now have some data that when we knock down STAT5, we can similarly prevent calcification in the dish. And really importantly, going back to those human tissues, when we screen, control, and calcified valve tissues, we can see TERT and STAT5 co-localizing in the sites near the calcification. These are serial sections here. So this black precipitation is where the calcification is, kind of showing up as this greenish background here. And you can see in the cells around that area, TERT and STAT5 co-localizing in the nuclei. Really importantly, you know, I mentioned at the beginning, the only therapy right now for calcific aortic valve disease is valve replacement. And a lot of the valves that we use are the bioprosthetic valves. That being said, they only last about 7 to 10 years. And this is actually an image of a failed bioprosthetic valve. This was extracted from a patient 7 to 10 years after the initial implantation. And really kind of one of the major goals in the field is to figure out how to make these bioprosthetic valves last longer. And, you know, thinking about the processes involved, you know, there are cells from the host that get migrated and grow on top, kind of an endothelialization of the bioprosthetic valve tissue. Well, we looked at some failed bioprosthetic valve tissues. These are 2 different ones on the top and in the middle here. And we can see TERT and STAT5 co-positive cells that are stuck on this calcified bioprosthetic valve. And so really what we think is happening is that there is an initial hit, an initial inflammatory event that is helping to trigger this cell to become a calcifying cell. And that initial event can stimulate the production of TERT and STAT5, which those 2 can then come together and help activate RUNX2, which is the master regulator of the osteogenic transcriptional program. And we've showed very clearly that if we knock down TERT, or if we knock down STAT5 or use a STAT5 inhibitor, we can prevent calcification in the dish. But what's the problem? The problem is STAT5, you don't want to inhibit that. right? These are people in their 40s, their 50s, their 60s, their 70s. If you give them a STAT inhibitor, that's going to have probably really severe consequences in their immune response. And so what we would love to do, oops, I'm going to skip over that. What we would love to do is figure out how TERT and STAT5 are interacting. And so to do that, we have gone to AI and used this really cool software called AlphaFold and the HDoc server. And these two things will help you predict the models of the proteins you're interested in. So we use the software to make essentially models of what the TERT and what the human STAT5 proteins look like on the structural level. And then using the HDoc server, figured out how they might interact. And so what I'm showing you here in blue is a dimer of STAT5. You can see the head here and the head here. It fits together kind of head to tail as a dimer. And then up here we have TERT. And when we flip this kind of like at a 45-degree, we can see the sites where TERT and STAT5 are interacting. And those correspond on this kind of linear map down here, where the TERT domain is in this RBD domain, which is the RNA binding domain of the enzyme, which is interesting food for thought. And then the STAT5 are kind of these, interstitial spaces that allow binding, but that change the conformation of the head of the STAT5 up here, which could enhance binding to the DNA. And so what we're working on now is making a system where we can actually visualize TERT and STAT5 interaction in real time. This is using a system called FAST, which was developed by a friend of mine, Alison Thibault. And what it is FAST is a very small but very potent fluorophore, and it can be temporally and spatially activated. And what we're hoping to do is use their system to understand TERT and STAT5 interaction. And so this is kind of the proof of concept of their study. They looked at FKBP12 and FRB, which only in the presence of rapamycin come together. And that's what their paper showed, is that they can very quickly, put these proteins together, add rapamycin, and things go away and things come together. And it's a very tunable system. And so what we're working on making right now is TERT and STAT5 connected to the N and the C terminus of this FAST system. And we're gonna have this fluorogen, so we can understand when and where in the cell these things are binding. We're seeing binding together both in the cytosol and in the nucleus, so we wanna watch those dynamics. But importantly, we want to make this system so that we can use it for drug screening, because you are able to fix the cells once you see it and then put them through a high throughput imager to see if, perhaps we can identify a novel compound that can inhibit this interaction. And so with that, I'm going to end. Hopefully we have time for some questions. This has been a huge effort in my laboratory. We've been working on this for several years now, and a majority of the work has been done by my postdoctoral fellow, Rolando Cuervas, also supported by many other members of my lab, Paria, Mark, Alex, Roman, and Michael, and also many former members. One, Luis, who was a former postdoc who actually started this work in the MSCs several years ago. And of course, my surgeon scientist collaborators, Dr. Ibrahim Sultan and my colleagues at Pitt and VMI and the Drug Discovery Institute, and of course, really most importantly, the donor patients and the families that enable us to do this translational work. So with that, I am very happy to take questions.
Hi, Cindy. That was an excellent talk. I learned a lot, and the tools you presented at the end were very helpful and might actually help stuff in my lab as well. So we'll go right into the questions. And the first question is from Corey Lau. Nice presentation. Do you think that TERT can inhibit calcium inhibitor, MGP, for example, as well, not only interacting with STAT5?
Yeah, so we actually have the MGP knockout mice, and we are trying to see if this is happening as well. And we do see in the MGP vessels, we only looked at the aortic root right now, but it does seem that the calcifying MGP knockout route has evidence of TERT and STAT5. Whether using a STAT inhibitor can prevent that, I don't know yet. We have not done that experiment, but it is interesting because MGP is, you know, it's a sink, it's absorbing, it's an anti-calcific molecule. So it'd be really interesting to see if TERT's playing a role, because we always think of MGP as an extracellular component. But stay tuned, maybe next webinar.
All right, next question is from Satish Srinivasan. Wonderful work, Cindy. Deepak Srivastava's group has reported that the deletion of TERT in Notch1 heterozygous background promotes valve calcification. How do you reconcile the differences, and what is TERT doing in a complex with STAT5?
What we think TERT, I'll start with the second one first. So I kind of skipped over it, but essentially we think TERT is the adapter between the chromatin remodeling components and the transcription factor that needs to get to the RUNX2. So I skipped over it, but we have some staining that shows TERT binding to BRG1. What we think is happening is that TERT is kind of that go-between. That is A, linking up these chromatin remodelers, and B, linking up the transcription factors necessary to drive transcription. So the first question though, how to reconcile that. So in the field, this Notch3 knockout mouse, it's really weird, right? Its penetrance is different. Evidence that it's actually active in humans is different. That's not saying their results aren't real. It's just saying that what we're looking at is only in human, and the mouse valve leaflet does not calcify like a human. And I think that's one thing we always have to remember. You know, we got to use the right tool to ask the right question. And that's partly why I'm starting with the human tissue, because mice are so tricky to calcify. You know, they are extremely resistant to calcify. And so what we see in them might be real for mice, might not be real for humans. And you know, hopefully, as more people enter the field and study calcification, we can really figure out the subtleties of these mechanisms.
All right, and I think our last question is, which TERT isoform is primarily involved? And then the second part to that is, did the STAT5 TERT also affect expression of other factors such as SOX9?
I forget. I don't think SOX9 was changed, but I think we pre-printed this, and the expression data we looked at is in that preprint, so it would be in there. I can't recall off the top of my head. You asked about isoform. So right now we have not looked at the isoform. The antibody we are using is specific for both. So we can't use that to determine which isoform. And then therefore we also haven't gone in and like tried to like, you know, CRISPR out one isoform or the other. So I at this point in time, I can't answer that question. But what I can say is the one thing we are very interested in is, you know, when TERT is interacting or is functioning at the telomere, it needs a long non-coding RNA to function, right? That's what TERC, T-E-R-C is. It's the RNA component that provides the primer to the telomere repeat. If you notice where our TERT and STAT interact, it is at that site. So one of the things we're actually interested in is the contribution of TERC-C, the RNA component, or are there other RNAs that might bind to TERT that help dictate its activity here? And we just don't know that answer yet.
All right, so looks like we're running out of time. So thank you very much for an outstanding talk, Cindy. It was really enlightening for me. And if anyone else has any other questions they want to ask, you can please email info@navbo.org, and we'll make sure to get your question to the appropriate speaker. So I hope everybody enjoyed today's webinar, and please look for more information about future events on the NAVBO website. And of course, we hope you can join us on June 1st when Dr. Gwen Randolph from WashU at St. Louis and Dr. Emerine Mughal from the University of Vermont present their lectures from the 22nd IVBM meeting. Thank you for joining us today.
Thank you so much, Josh. That was great.
Thank you, Cindy.